So the second talk will be given by Sandro. So sorry, I will turn on my video. Yeah. Okay. So Sandro is an assistant professor of neuroscience in the Mayo Clinic College at Jacksonville in Florida, and as a postdoc in the lab of kidneys and as a new PI, he has been studying the role of meningeal lymphatic system in both brain physiology and brain pathology. Of course, with the objective of finding new therapeutical pathways to explore in neurodegeneration. So, and today he will give us an overview of his work, and with a special focus on lymphatics, meningeal lymphatics, and the role in Alzheimer's disease. So, and I would like to remind the audience, if you want to ask questions, please just type them in the box. Thank you. Please, Sandro, the stage is yours.
Okay, thank you, Emanuela. This was a very nice introduction. I'll share my screen. And while I'm doing that, actually, I want to acknowledge all the work from Jerry Lin and all the folks behind the Open Box Science, because this has been a really nice initiative, and I've been attending some of the talks, and it's been really cool. So as Emanuela was mentioning, today I'm gonna briefly go through main findings of two of our recent studies. But before I jump into that, I think it's important to take a step back and just talk a little bit about, or give you some background about meningeal lymphatics or lymphatic vasculature and its role, and also some of our previous findings that really paved the way for these new studies and experiments that we designed in these two new studies. And actually, let me just change this to laser pointer. So I think by now it's not really extremely novel that we do have bona fide lymphatic vasculature in the meninges. And let me just take this bar out of the way as well. Okay, now it's better. And these lymphatic vessels, so they express all of the characteristic markers of lymphatic endothelial cells like LIVE1, PROX1, VEGFR3, and others like CD31. And these lymphatics, they're located alongside the main blood vessels of the meninges, okay? And this is true both for rodents, but also for larger mammals like macaques and humans. So it seems to be a pretty conserved system. And it was recently characterized. So we're still trying to figure out, its structure, how it develops, its functions, and while we're doing that, we know for sure, or some studies in the recent passage have shown that this type of lymphatic vasculature in the meninges depends on constant signaling by VEGF-C through VEGFR3 receptor. And it's necessary both for the development of meningeal lymphatics, but also for their maintenance, okay? And these lymphatics, morphologically, they are arranged as initial lymphatics that are not as restrictive to passage of both molecules and immune cells into the lumen of these initial vessels. And then they converge into larger collecting-like lymphatics. And then these exit through the skull foramina into the peripheral environment, and then can converge into what traditional collecting lymphatics that have smooth muscle cells and valves. And these guys, they are draining into the peripheral cervical lymph nodes. And what is really interesting about this type of vasculature is that it's constantly draining CSF molecules or CSF contents as a whole, and they're also involved in modulation of several immune cell population egress into peripheral lymph nodes, and this obviously follows more regulated mechanisms, such as, for instance, signaling through CCR7. And what we really know due to recent work is that at particular anatomical locations within the meningeal compartments, both molecules that are present in the CSF or in brain fluids and immune cells of the dural compartment, they do have access to the lymphatic vasculature. And if we're talking about molecules, there's different ways to assess this process. We can inject either fluorescent tracers with different sizes into the CSF, and then follow their drainage into peripheral lymph nodes. And what we realized was that in terms of molecular drainage, obviously for immune cell, the dynamics of the process is different, but for molecules that are present in the CSF, this drainage is happening actually at a quite fast pace. So this is a live video showing fluorescently labeled microbeads being drained from the CSF through lymphatics into the peripheral deep cervical lymph node. And what we also know is that in healthy conditions, or the healthy brain is devoid of lymphatic vasculature. So at the level of the brain parenchyma itself, we have other systems, other structures that are involved in clearance of the brain and cleansing of the brain. And actually what we asked ourselves initially, and this brings us to our previous studies, is whether if you have a dampening of lymphatic vasculature, how does this affect either other pathways of brain cleansing or several different aspects of brain physiology and function? And I'm just going to go very fast through our previous findings. So in these previous study, we used different models of loss of function of meningeal lymphatics. One of those models was the photodynamic ablation using Visidim. And that was, as you can appreciate here with these images, we're able to efficiently ablate lymphatic vasculatures marked with live one in green, but we are sparing blood vasculature. And this resulted in a significant decrease in drainage of CSF content. In this case, our surrogate was fluorescently labeled ovalbumin into deep cervical lymph nodes. And what was really interesting is that if we induce this ablation, this meningeal lymphatic ablation method, we observed a great reduction in another system that is involved in brain cleansing and fluid recirculation, which is the glymphatic system. And not only that, but mice with impaired meningeal lymphatic drainage, they also showed this mild cognitive impairment phenotype in the Morris-Water maze, which is a test where mice have to find a hidden platform using spatial cues. And this kind of gives you an idea about their learning and memory capacity. And we also observed that with aging, you actually have these a very overt changes in morphology of meningeal lymphatics. You have decreased coverage, and they also have decreased capacity of CSF drainage. So there's this impairment of meningeal lymphatics with aging. So we asked, okay, can we provide something to improve this? And remember, I told you that VEGF-C is one of the growth factors that is very important for lymphatic maintenance and function. So we provided VEGF-C to old mice lymphatics, and we actually were able to partially rescue their morphology and also their drainage capacity. And this resulted in improved cognitive capacity of older mice. This is what I'm showing here is, again, results in the Morris-Water maze. And this was actually achieved specifically through lymphatic drainage, because if you ligate the collecting lymphatics at the deep cervical lymph node level, you actually abrogate this beneficial effect of VEGF-C. And what we've shown using Alzheimer's disease transgenic mouse models that mimic, that increased amyloid deposition in the brain, is that if you induce meningeal lymphatic malfunction, you're actually going to aggravate amyloid accumulation in the brain. And not only in the brain, but to my point, even more surprising, in the meningeal compartment itself. So if you look at an adult 5XFAD mouse, you don't really see much amyloid deposition. But if you ablate lymphatics, now you start to see what we call the meningeal amyloid angiopathy. So a lot of amyloid starting to deposit on meningeal blood vasculature. And this actually resembled a lot what you see in AD patient dura, okay? So as you can appreciate, if you look into meningeal tissue from Alzheimer's patient, there's a lot of A-beta accumulation. And we actually were able to mimic this in the mouse model, in an adult mouse, at least, when you dampen meningeal lymphatic drainage. So this brings us now to our new studies, okay, newly published studies. And in this study, it was one of the questions in this particular study that we published in Science Advances, one of the main questions that we had, okay, so lymphatic drainage has this dual role. So they are draining both molecules from fluids and from the meningeal compartment, but also modulating immune response, okay? So we asked ourselves, okay, can just that particular harm of crosstalk with the immune system be responsible for some of the phenotypes that I've just showed you, both in aging and in AD using mouse models? And initially we decided just to look, since we knew that lymphatics, they start to malfunction with aging, okay, let's look at the immune response in the meninges, at the immune system in the meninges, and how it looks with aging, right? How it changes with aging. And it's really well documented that in aging in the periphery, you do see changes in immune response, in immune system. At the meninges, it's very consistent that we see higher numbers of T cells, okay? So there seems to be some T cell accumulation in the meninges of old mice. And what is interesting is that if you look at CD4 T cells, so, and this goes for CD4s and CD8s, if you look at CD4 that express FOXP3, so transcription factors, so Tregs, there seems to be an increase of this particular flavor of T cells, the Tregs. So, okay, we ask ourselves, can this be somehow, these changes with aging somehow related with different immune cell egress through lymphatics, as I'm showing here in this diagram? And one of the mechanisms responsible for this is CCR7 expression on the immune cell itself. So we decided to measure CCR7 expression in old mice. And as you can appreciate, in different T cell populations, you actually see this decreased CCR7 expression by T cells. So, okay, is this somehow related, right? Is this decrease in CCR7 leading to this accumulation in meningeal compartment? So in order to answer that, we used bone marrow chimera models. So we either replenish wild-type bone marrow with wild-type bone marrow as control, or CCR7 bone marrow. And what was actually interesting is that if we do so, we are able to recapitulate what we see in an old mouse. So if you look at meningeal immune cells, you see, again, this increased number of CD4s and CD8s, and actually you see the opposite in the draining lymph nodes, right? In the deep cervical lymph nodes in this case, less CD4s, less CD8s. And this was really interesting because we wondered if you can use this model again to disentangle what is the influence of lymphatic drainage on the CSF composition and CSF drainage, or modulation of immune cells. And what is interesting is that if you compare CCR7 knockout mice with just regular wild types, they do seem to present very similar, or we couldn't detect any differences in lymphatic morphology, and they do have the same capacity, similar capacity to drain CSF content, okay? So it seems like that part of lymphatic function is not affected in this model. However, if you look at the immune system at the level of the meninges in these mice, you do see those changes that I just described, right? And this is obvious if we do a more in-depth phenotyping using Sci Adv, for instance. So you start to see a lot of these FOXP3 high populations, and this is quantified here. And what is interesting is that although you see increased numbers of CD4s and CD8s, they seem to be expressing lower levels of, so you see less Tbet high cells, so less amount of cells that have the capacity to produce interferon gamma, okay? So there seems to be this more immune suppressive environment in the meninges of CCR7 mice, okay? So we wondered, okay, are the phenotypes that I've just showed you before in models of lymphatic ablation also recapitulated when you had this loss of CCR7 expression on immune cells? And the answer is actually yes. And this was also very surprising. If you look at lymphatic function, it's also affected in CCR7 knockout mice. And if you look at learning and memory in Mori's water maze, again, constitutive CCR7 knockout mice, they do show, and actually we also observed deficits in novel location recognition test. But in both tests, you see that mice show this mild cognitive impairment. So they show worse performance in NLR, they show worse performance in Mori's water maze. And this seems to be specific to loss of CCR7 in the hematopoietic compartment, because again, in our bone marrow chimera mice that have received CCR7 knockout bone marrow, they also show this mild cognitive impairment. Okay, so let's look at AD transgenic mice. So these mice, using CCR7 reporter, we crossed these mice with CCR7 reporter mice, and we observed that these mice, as they age, right, comparing 3 months and middle-aged 5-XFAD, they show also this decrease in CCR7 expression at the level of the meninges and also the draining lymph nodes. And if you cross 5-XFAD mice with CCR7 knockouts, you see this aggravation of amyloid deposition in the brain. But what is interesting, and actually we include in supplementary data, is that you don't really recapitulate all of the aspects of meningeal lymphatic ablation, right? Because if you look at the meninges, you don't recapitulate that overt increase in meningeal amyloid angiopathy in the CCR7 knockout mice. So not all consequences are the same, but it seems that you do recapitulate some of the features of meningeal lymphatic ablation in mice that have reduced expression of CCR7 in immune cells. And now we wondered, okay, can we improve or rebalance the immune response at the level of the meninges or peripheral immunity in old mice, and this way improve their brain function, right? So we decided to treat old mice with anti-CD25 because we know that FOXP3 positive CD4s, they express high levels of CD25. It's also highly expressed in effector T cells as well. So we treated these mice with anti-CD25, and we test their function again in Morris-Water maze and novel location recognition test. And these mice are performing a lot better, okay? So this was really interesting. And if you look again at their immune response, these mice, they also, alongside with the better cognitive function, they also show less of these Treg population in the meninges and also in the cervical lymph nodes. Although using this approach, we didn't really mess up with the amount of T cells, just the flavor of the T cells. So this was, I think, a very interesting study altogether, and these findings seem to go along very well. And we included this scheme in the paper as well, which kind of sums up main findings. So basically, if you have this age-related CCR7 deficiency in immune system, which CCR7 is expressed in high levels in T cells, you start to see impairment in lymphatic function, in vascular function. I didn't show much of these results, in amyloid beta pathology. And then if you try to rebalance this in old mice with anti-CD25 therapy, you actually reduce this Tregs in the meninges and also improve the cognitive function of aged mice. So this is one of our recently published studies. The new study, the other new study that I'm presenting today, it basically focuses more on lymphatics and modulation of brain parenchymal cell response, and also response to treatment in the AD context. So what we're initially showing in this paper is that if you compare, so if you let 5-XFAD, so AD transgenic mice, if you let them age, and you look at their lymphatic morphology, you actually see, alongside with increased A-beta deposition in the meninges, you see this aberrant lymphatic morphology, so decreased coverage of lymphatic vessels. So we wondered, okay, if you have an AD patient aging, right, and their lymphatics start to malfunction, can this have an impact on, for instance, innate immune response in the brain, and also in therapeutic strategies that are being developed? And one of them is immunotherapy. Okay, so we asked a very simple question: does meningeal lymphatic dysfunction affect immunotherapy against amyloid beta? And in our experiments, we used both the murine version of aducanumab and BAN2401, which are two anti-amyloid antibodies, monoclonal antibodies that are being tested in clinical trials. Aducanumab was actually recently approved for treatment of AD patients. And we focused on different crosstalk aspects that could be changed by the crosstalk between lymphatic drainage and immunotherapy, being one of those A-beta plaque clearance, and also neurovascular and microglial response in these mice. So this is experimental design. So we used our photodynamic ablation model to induce lymphatic malfunction, meningeal lymphatic malfunction specifically. And we treated mice with aducanumab or IgG isotype control. And what we observed is that although we don't really affect, so mice with impaired lymphatics, they don't really show decreased efficiency of aducanumab, but they do show a bigger amyloid burden at the end stage of the treatment, right? So, and this is very obvious here by the images. So mice treated with aducanumab that have intact lymphatic drainage, they show this overt reduction in amyloid burden. Mice with impaired lymphatics, they show more amyloid, but then mice with impaired lymphatics that are treated with aducanumab, they show a little bit more amyloid than these guys with intact lymphatic drainage. But what was really, really interested, even more than amyloid clearance, was activation of the innate immune cells in the brain, the microglia. So regardless of being treated with antibody, with aducanumab, the microglia, they show the overt activated phenotype in mice with impaired lymphatic drainage. And we also see more fibrinogen accumulation in these mice, which suggests some sort of vascular impairment as well. So in order to delve more into the response by brain, blood endothelial cells, and also microglia, we performed single-cell RNA-seq after enriching these cells for blood endothelial cells and also myeloid cells. And if we focus on the transcriptome of capillary endothelial cells, you're able to see that there's upregulated response to damage, different types of pathways involved in that. And also we see a downregulation of immune signals that might be important for vascular function. And we see, for instance, the response to interferon gamma is down, response to chemokines is down. And we wondered whether, microglial response could kind of go hand in hand with this. So microglia, they showed pathways that are involved in an activated phenotype. So it seems like they show this exacerbated activation, which might be deleterious in the context of immunotherapy, for instance. And what is interesting is that they showed downregulation of a lot of pathways that suggest a dampened crosstalk with the adaptive immune response arm. So for instance, decreased T cell activation pathways and antigen presentation. So we wondered if these transcriptomic changes in microglia in the mouse model in response to loss of meningeal lymphatic drainage could somehow have show a parallel to what's happening in humans, in the human brain. So we performed an integrated analysis of human and mouse microglia, transcriptomes. And for that, we collaborated with the group of Oscar Harari and WashU. They are doing a beautiful work in human microglia transcriptomics. So we decided to look into our data sets in an integrated way. And as you can appreciate here, we doing our integrated analysis, we can identify a homeostatic microglia signature, let's put it like that, a more activated one, which is cluster 2 here in red. And then you also observe what we call these alternatively activated microglia, which we don't really know what they represent in terms of exacerbation or inflammation in the human brain. And these in cluster 2, they showed like a dam-like signature, let's put it like that. So the disease-associated microglial signature. And when you look at our 5XFAD microglia, you see that they kind of go, they follow into different clusters. But what is really interesting is that if you compare mice with ablated lymphatics, right, and mice with intact lymphatics, with our different microglia coming from different patients, is that mice with intact lymphatics kind of resemble the activation status of microglia from non-AD patients, and our mice with ablated lymphatics, they are transcriptomic signatures. They follow more into the sporadic AD or AD cases in general. So this is really interesting, and we actually devised a signature of microglia from mice with ablated lymphatics and showing similar results as the previous graph plot that I showed you. This signature falls within the activated microglia from cluster 2, 3, 4, and 5. So it seems like mice with ablated lymphatics the microglia activation status resembles more that of AD patients than compared to non-AD patients. So in order to understand if not only if ablation of meningeal lymphatics has a function, but improvement of meningeal lymphatic function leads to better action of anti-amyloid beta antibodies, we treated old AD transgenic mice with viral vector that expresses VEGF-C in combination with our anti-amyloid beta antibodies. And in this case, we see that there is a even better reduction of amyloid beta from the brain of these mice. And alongside with this improvement in antibody efficacy, we also see that there are changes in microglia and also in vasculature that are in some way opposite to our paradigm of meningeal lymphatic ablation. Now you see that there's more interferon gamma and also interferon signaling in these cells, and this might actually be in this context of immunotherapy beneficial for removal of A beta and potentially to other aspects to improve other aspects of brain function. And this is basically just a sum up of what I've been telling you, and also a sum up of the status of the field right now, where you have a lot of other groups delving into meningeal lymphatic function, not only in the context of AD, but also in context of Parkinson's, traumatic brain injury, subarachnoid hemorrhage and whatnot. And it seems like lymphatics, they do need to be considered both as a new system that we have to consider in terms of therapies, but also in terms of potential disease pathophysiology. And with this, I would like to thank a lot of people, because this was a lot of people were involved in this study, especially Yoni Kipnis, which was my postdoctoral mentor and supervisor in many of these studies. A lot of people in the Kipnis lab, collaborators, our new collaborators now that I opened my lab at Mayo Clinic, collaborators from other entities and also from Mayo, funding agencies, and also my beautiful lab members, current lab members who are doing a lot of hard work now in the meningeal lymphatics and neurological disorders lab at Mayo Clinic. And thanks for your attention, and I'll take questions now.
Thank you, Sandro. Thank you for your really nice talk. So we have some questions from the audience. So there is one question from Eugenio Contreras. Yes, any crosstalk between Tregs and gamma delta T cells in the meninges in your model? I think this refers to your first part.
Yeah, that's a very good question. So we did quantify differences in gamma delta T cells when we compared middle-aged 5XFAD and adult 5XFAD. There's no changes in cell numbers. Well, that doesn't mean that there's no differences in crosstalk, obviously. And this change in Tregs, so the increase in Treg frequency and number with aging, we do see them in the 5XFAD model as well. So short answer is I don't know if there's any crosstalk, but it might well could be any crosstalk. Yep, because they are very close to each other, right? So T cells, they are all along or mainly located along the dural sinuses, right? So yeah, they can easily interact with each other and secrete factors that influence each other's function potentially.
Then I have a question from Lynn. Why do you think there is no beta deposition in mouse meninges?
Oh, why? Because I think they have intact lymphatics, right? So that's what we're showing. So we compared lymphatic function between an adult 5XFAD and wild-type littermate control, so hemizygous non-transgenic mouse. They have this exact same lymphatic morphology, meningeal lymphatic morphology and drainage capacity, right? So only when these mice age, you start to see that the lymphatic vessels deteriorate, right? So that's why I think if you look into these AD transgenic mice when they are very young, they're lacking that lymphatic malfunction that you only see with aging, right? And that's why, with our manipulation, with our loss of function models, we were able to recapitulate that pathological feature that, it might well play a role in disease, right? Because you see that pathology in AD patients, in AD patient meninges, right? So.
I have another question from Wei Chao. What do you think of VGFC for a potential human treatment to boost the meningeal lymphatics?
Sorry, to the VGFC for?
For a potential human treatment to boost the
Oh.
meningeal lymphatics.
Oh, what? Well, you know, it's very risky to say, oh, yes, this is going to be a very promising therapeutic strategy. I mean, we don't know at this point, right? Using the mouse model and our readouts, right? It's all, you always have to take into account that we can only go as far as we can with readouts, right? And we looked at amyloid clearance or amyloid levels in response to immunotherapy alone or in combination with VGFC, and it seems like antibodies are able to clear plaques better in very aged AD transgenic mice, right? It seems like in old mice, if you increase VGFC levels at the level of the meningeal compartment, you also see beneficial effects in terms of lymphatic drainage and cognitive function, right? Can this be a very promising therapy for human condition? I mean, based in our results using mouse models, it seems so. But we have to do a lot more experiments and have a lot more controls, and then we need to go into clinical trials or whatever in the future to be really sure that this is a promising strategy to follow. Because in some other contexts, increasing VGFC, for instance, in certain cancer types, it was shown by others that it doesn't have beneficial effects. So it has to be context dependent and situation dependent, right? But I like to believe that it should be considered at least as potential therapy for this type of aging related disorders. Yes.
Yeah. So I have a couple of questions from Luca De Feo. So apparently in AD mice, we don't have interferon gamma dependent inflammation, and increasing Tregs leads to anti-inflammation. Could it be that this effect is masking a beta phagocytosis and removal?
Yeah. I mean, that's, it seems like with aging, we do have this more at the level of the meninges and the meninges deep cervical or cervical lymph node axis. It seems like you have this more immune suppressive response. This might not favor brain resident innate immune response, right? Because if you need them to be active and to phagocytose and to remove waste from the brain, having this more immune suppressive environment apparently might not be beneficial. So that's why we think that, for instance, with anti-CD25, it's like giving a rebalance in adaptive immunity that might favor then innate immune response at the level of the brain parenchyma. So yeah, it might well be that having immune suppression at old age is not the way to go, or at least you have to have some rebalance of immunity, and that's what I think we're achieving in our mouse models, and that's why it's showing beneficial results.
Yeah, and then there is a second question. Could it be possible to link a beta to some soluble oligomers to promote its own drainage?
Right. Yeah, I had a similar question before. With the A beta antibody complexes, if they are drained by lymphatics, this is kind of going in the same direction. Yeah, I mean that's why we think that it's not enough to have a good antibody that removes or helps to clear plaques, right? Or those bigger clumps of A beta in the brain. You also need to have a good system that then takes all of that waste out, right? And then obviously BBB and blood vasculature will most likely play an important role there. Brain phagocytes will also play an important role there, but I think lymphatics will also be very important, right? If you don't have a functional lymphatic system that is constantly draining the brain, you won't be able to remove these A beta antibody complexes out of the system, which at the end of the day might also cause some damage. Yeah. But we don't have experimental data to support that at this point, right? But we do believe that A beta as a soluble molecule will behave as ovalbumin any kind of tracer that you inject or molecule that you inject in the CSF, and will eventually be drained by lymphatic vasculature.
Thanks. So I had a question actually. From your single cell data on the CCR7 knockout in the meninges, you show an increase. So you show a decrease in the Tibet positive population, CD4, but you also have an increase in the double negatives. So do you know which would? So we also see that a lot. So I was wondering if you know what this would be.
You're talking about double negative T cells in the meninges?
Yeah. Exactly.
That's a very good question. I don't know what those are.
Yeah,
I remember. Yeah, I remember. I'm not sure. I mean, if you take out, when you're analyzing your T cell compartment, if you take out the gamma deltas, right? Well, but those were TCR beta. Yeah, I mean, I had some discussions in the past with other colleagues about those double negative. But I don't know if that's real, if that's something, you know. I don't know. I prefer not to comment on that at this point. Yeah, but it might be interesting to delve into those double negatives to see if they're really doing something.
Yeah. So, and then I left the best question for the last. So could you share your secrets to be a super productive scientist?
Oh, well, I think, you know, I don't think there's a, you know, a huge secret. Is that you just have to really enjoy doing science and doing research. You have to be motivated every day. It's not easy, right? It's 95% failure, and if you're lucky, 5% success. Maybe the percentages are even lower, I don't know. So you have to be motivated, and, you know, have to be curious constantly. You have to work hard. You don't have results if you don't work hard, and you have to achieve a very good work-life balance. I mean, it's not easy. I mean, it's not easy, but it's possible. It's possible, and if you like what you do, then it's even easier, I guess.
Okay, thank you so much, Sandro, for the nice talk.